Okay, I guess we'll get started with the second lecture. So thanks for Professor Pankaj for the first lecture, which actually introduced a lot of great fundamental ideas, which we'll use later on. So I'm going to, the first lecture, for me, I'm going to give a brief introduction to active systems and talk about sort of common examples that people have used throughout the years. One is to understand how they behave physically, but also the context is to think about what does that mean for biology and for engineering and other applications. So when we talk about active matter, we typically talk about, as Professor Misher has said, we talk about the system, which are made of particles. So notice that particles here are in quotes, so they don't have to necessarily just be particles. They can be any kind of entity or object in the system. So the key idea is that these particles, they all consume energy, whether that's stored by themselves or ambient energy from the environment. So they use that energy to do two things typically. One is to generate some kind of motion. So if we're thinking about humans walking around or bacteria, things like that, or other systems that generate mechanical stress, which we'll talk about later on, such as things like microtubules, which are systems in a cell. So if we think about a passive system, we have, for example, particles here. They're not injecting energy at the individual particle level. And for active particles, they're because they're consuming energy and they're generating motion constantly. And that means they're inherently out of thermodynamic equilibrium at all times. So there's always an energy injection at each particle level. So that's sort of the key difference between an active and a passive system. And because when we're talking about active systems, typically we're talking about multi-particle systems, right? So there are a lot of, I think this one is off for some reason. Okay, it's back. So this system is made up of a lot of individual entities, and they often have all kinds of interactions, whether that's chemical, physical, or other rules-based interactions. So these interactions often leads to emergent collective phenomena at the macroscopic level. So here are a few examples of physical interactions. For example, you can have excluded volume interactions for particles this looks like elongated particles. You can have short-range attractive forces that will lead to interesting phenomena, which we'll talk about later on. And you can also have different kinds of collisions that depends on the context. You can make up different rules to describe their interaction. For example, you can have inelastic collisions or collisions that are nematic in this fashion. So let's look at examples of active systems where the individual entities are large or macroscopic. So here are a few examples. The first one is starling. You can see these frogs, and you can also have fish schools. Okay, these are all particles that are sort of macroscopic level. You can see these individual particles with your naked eye. Here's an interesting video of a flock of sheep going through a narrow gate. So what you see here already is there's an interesting pattern that emerges. So if you look at the density of the sheep, what you see is near the gate, the density is higher, and that's actually a very important phenomenon that we'll talk about later on. Whereas if you're far away from this gate, you see that the density is less, and also there's spatial inhomogeneity that develops. And another example that's quite interesting is that of the army ants. So they form this sort of interesting state where what happens here is these ants, so each ant follows the one before it, and there's an instability that occurs where they form into this vortical state because they're just following the one before them. Eventually, what you see is many of the ants in the middle have already died because of exhaustion. So that's a very cruel reality of collective phenomenon for ants. Okay, so as humans, we're also active systems where each of us is an individual active particle. And here's a video taken in a crosswalk in Japan, which is one of the busiest crosswalk. So what happens here is every about 2 minutes, there's more than 2,000 people that goes through this crosswalk. So as humans, we're active systems, and we can make use of these ideas to understand, for example, crowd dynamics in the context of, evacuation modeling and things like that. So that's also something that people are starting to think about at this level. So let's go down the length scales and now look at systems. At smaller scales, so at colloidal scale, which means individual particles now are about a micron in size. So a prototypical example is motile bacteria, such as E. coli. So here's the experimental video taken just putting E. coli in water, so they're swimming around, and this self-propel. Later on, we'll talk about how these organisms self-propel or how they generate their motion. Here's another interesting microorganism. So these are a type of green algae called volvox. So they have these spherical colonies. What happens here is because of the hydrodynamic interactions between these colonies, they form these interesting bound states where you see these little colonies are rotating alongside a bigger colony. And what's perhaps most interesting in the last decade, where active matter really become an emergent field of study, is because now we can easily make synthetic active particles. So these are just colloidal particles. So what these are is really they're just plastic beads. And what you can do is you can coat these plastic beads with catalyst on one side of the sphere, say only half of it, and you can put that into a hydrogen peroxide solution. So this catalyst, promotes the decomposition of hydrogen peroxide, and that generates a chemical asymmetry because only one side of the particle has a catalyst. So that allows these guys to self-propel. And just due to the fact that these guys self-propel, what you see is they self-assemble into these interesting structures. Okay, so there's no attractive interactions among them. They're purely repulsive in this context. And in this particular experiment, what you can do is you can make these sort of particles light responsive. So if you have light, if you shine light on them, the chemical reactions is going to happen, and then they become active. But also, as Professor Mishra said, if you turn off energy injection, if you turn off light, if you turn off chemical reactions, what you see is these clusters dissolve away just by diffusion, and then they will eventually relax over time. Okay, we'll even go down a smaller length scale to look at active systems in cell biology. So these are very interesting systems for biologists. For example, on the left, I'm showing this is a artist's depiction of what's called the inner life of the cell. So this is not a real video, okay? It's made by an artist, but it does visualize the essential physics that's happening here. So what you see here is there's a large vesicle, okay?
Look at this one.
And you see these tracks. So those are called microtubules. So those are essentially highways within our cells. And what happens is, so this little guy here, that's a kinesin motor. It's a motor protein that walks on the track. And by walking on the track, it's going to drag along this vesicle. So it transports materials from one part of the cell to another. That's very important for cell functions. And one might be wondering, why can we simply rely on bulk transport? For example, why don't we rely on diffusion for these processes to happen? One reason is the cell is a very crowded place. If you want to rely on just bulk transport to move these vesicles, it will be virtually impossible for cells to function properly. That's why they have these microtubules as highways in the cell. Okay, so that's a very interesting active system. And similarly, on the right, what I'm showing is a video about cytoplasmic streaming. So what happens here is you see that there's a systematic flow of the cytoplasm within a cell, and these green organelles, so these are sort of membrane-bound organelles. Those are chloroplasts. Those are where the photosynthesis happens in the cell. So what happens in this case is, because of these kinesin motor, or the proteins that are sliding on these filaments, they will generate these active forces within the cell, which generates flow. And that's also interesting for cell function. Of course, much of these phenomena we know, but the point is, how do we understand these systems from a more physical point of view? That's still at its infancy at this point. Great, so the point is, why do we want to study active systems? Well, from a physics point of view, we want to study active systems because it's a sort of perfect platform for developing non-equilibrium physics. And the other interesting thing in connection to living systems is, we know that physics is very good at describing non-living systems. And the question is, can we do the same thing for living systems? Can we develop a very systematic tool and framework to understand living systems, such as for the context of biology and things in general? Yeah.
In the previous slide,
Yeah.
for the protein case, what is the constituent particles of the active system?
So on the left,
Yeah.
so that would be the motor proteins, and it's actually a composite system, which I'll show you later in a moment. But essentially, so these motor proteins will bind to the microtubule, And in this case, so this guy doesn't so actually what happens is, if you're a biologist, you can take these entities out and you can sort of cross-link two microtubules with the motor protein. And in that case, this aggregate is not self-propelling, so it's not generating net motion, but it's only generating stress. So in that case, that's the second sort of class of active matter where there's no self-propulsion, but there's force generated, which means that's going to generate flow in the fluid. Yeah.
Because I was still confused because for all the system waves,
Yeah.
the constitutive particles are there are so many of them.
That's right.
Just we want.
No, this is we're only showing. So this is the artist's visualization. Of course, there's many of them that's happening in the system, but yeah, exactly. Yeah. Yeah. Right. Yeah. So that's a passive load essentially that's being carried by this protein. Yeah. Other questions? Okay, great. So we're talking about why do we want to study active systems? From a physical point of view, we want to develop non-equilibrium physics to describe these systems, but also we want to understand living systems under the same framework of physics that we're familiar with. And of course, there's lots of applications. I've shown you a few examples already. One is sort of cell biology. How do we understand fluctuations within the cell environment, and how does those promote transport processes, which are important for cell function? And other interesting aspect is in engineering, where we want to make micro or nano robots for things like delivery and sensing tasks. So here's an example where people have made a nano robot using a platelet cell that's coated with a natural protein. And this will catalyze a reaction that's sort of already happening within the human body. So this would be an example where you can make use of chemical reactions, and then you can allow this guy to self-propel, so which we'll talk about later on in the context of drug delivery. And a third example is in materials science, because these self-propelled particles, they form interesting structures, so they're useful for self-assembly. And the other aspect of that is we can sort of add a fraction of active particles in a passive system, such as in a gel. And what happens is, if you dope a gel with active particles, you can actually modulate the physical or the mechanical properties of a gel with only a small fraction of active particles, which we'll show an example later. Okay, so any questions about this part? So because there are many active systems, and we can come up with ways to classify them. So here's a few examples. The one I already showed is look at the length scale of the individual particles, right? That's important for a reason which we'll talk about later on. You can look at the origin of their self-propulsion or activity. We have biological systems like microorganisms, humans, animals, or you can also have synthetic particles that self-propel through a chemical mechanism or another mechanism. And you can also look at the symmetry of motion, which means whether there is self-propulsion. So for most of the examples we looked at earlier, these are all self-propelled, so they generate motion. So those are called polar systems. But there are also pneumatic systems, which we'll talk about in the context of the motor proteins. And I can also look at the environment. So there's sort of dry active matter. For example, if you're thinking about humans walking on the street, that's a dry system, so there's friction. And you can think about the wet active matter, which means just particles or entities in water or even in a liquid environment. So there are many other ways to classify these systems. This is only a few examples. And so let's look at a few model systems that over the years people have been using to understand active matter. So I talked about this motor protein system. So what happens here is we have these microtubules that I showed earlier in the video. So these are about 10 micrometers in diameter, in length. So this microtubules has what's called a minus end and a plus end. And that has implications for the motor protein. So when the motor protein sort of combines with the microtubule, it walks from the minus side to the plus side. So then in this case, it will drag these two microtubules towards the center, right? So in this configuration. And notice that if you look at this whole system, which is made up of these two microtubules and the motor protein, there's no net self-propulsion that's been generated. However, there are active stresses that happens which will disturb the fluid. And if you're a biologist, what you can do is you can make these motor proteins light responsive, okay, by doing bioengineering. So essentially, in this case, this motor protein is made up of two parts. If you shine light in the system, it will aggregate, form a functioning motor protein. And if you turn off the light, they will dissolve, and then they cannot cross-link and walk on the microtubule. And so in this case, if you take a purified microtubule solution with motor proteins and ADP and their energy in a solution, if you shine a light pattern here, in this case, that's a disk pattern, what you see is these motor proteins will aggregate and they will cross-link with microtubules, and that will aggregate these microtubules in the light pattern, right? So that's what happens here as a schematic. So if you look at an experimental video, so here's a disk pattern of light, and what you see here is now, so the bright spot, that's the density of the microtubule. So initially the light pattern is turned on. Let's watch that again. So the light pattern is on, and then these motors are going to aggregate microtubules, and that's why you see a bright field there. And then the disk pattern is, the light is turned off. Again, you've turned off the source of activity, so then this whole system will dissolve away, and then you form a homogeneous state in the experiment. And here's a second example where you have a bar pattern of light. Again, you are going to aggregate microtubules, but in this case, you can sort of turn the light pattern in a rotational fashion, and what you see is you generate this disturbance flow around the system. I guess those are hard to see here, but the point is you can sort of, what you see here is there a flow that's coming in because of the aggregation of the microtubules, and you can sort of modulate the flow environment at this level by changing the light pattern. So let's see this again. We can see the flow field here. I guess you guys can see the tracer field, right? The gray ones, those are tracers where you can see the flow field, but anyway, that's fine. I'll share this later on. Okay, so that's microtubules, but you can make more complex microtubule networks. For example, here, previously I talked about two microtubules that's linked by proteins, but then you can form a network. So you can have a third one with other proteins that form the longer and longer network. So in this case, you can imagine you can then form a very dense network of microtubules, and then they can generate spontaneous flow in the system due to instability. And so these are typically called pneumatic systems, which we'll talk about later on. And here's another interesting experiment.
It is in vitro experiment.
Yeah, so there's a taking out taking out the microtubules and motor proteins into a lab environment. Yes. This one doesn't show.
So light is the source of activity in vitro.
Sorry?
The experiment in vitro.
Yeah.
Light is the source of activity there.
Yeah. So in the previous, yes. So the light sort of controls whether the protein can function or not. But there's energy. There's ADPs in the solution that they consume as fuel. But they use light as a way of whether the activity is present or not, by sort of directly tuning the self-propulsion mechanism or the activity, the origin of activity, right? But the energy is there supplied in the solution. Yeah.
Energy is being supplied by the light.
No, it's in the solution. The light only controls the.
It's coming from the solution.
Yeah.
Light is free.
The light is so the motor protein has to bind to function. So if you don't have light, these proteins will sort of disassemble. They will unbind, and then they cannot cross-link with the microtubule.
So what is source of that in vivo?
Sorry?
And inside the cell.
Yeah. No, that doesn't happen in the cell. So that's done by genetic engineering. Yeah. It's not. It doesn't naturally happen. Yes, in the cell, the proteins are always properly functioning, But in an experiment, that's why we can control them that way by sort of genetic engineering these motor proteins.
Like how? Just one. So motor protein is bringing all these microtubules compartment like their structure.
Yeah. So the microtubules, if you're on their own, they're just passive rods that are
Protein molecule is fully
So they will So the motor protein has two ends.
cross
So
linking each microtubules with each other.
yeah, and then they're gonna start walking on the microtubule.
And then they wipe out.
They walk on the microtubule, and then they will sort of these guys will slide along right in opposite directions.
Is here protein molecule?
It's a composite, right? Yeah, so it's the motor protein microtubule composite. Yeah, as a sort of the particle in the active particle system. Yeah.
I mean, what need them come together that orbitals?
You mean this one here?
L02 is fine.
Yeah.
But all others. Means when they are coming together.
Yeah.
That's all.
Yeah.
So it is being done by the protein molecules or microtubules are also interacting with each other.
There's in this case, in the network case, no, there's no direct interaction between the microtubules. They're only sort of connected by the motor proteins, and then you form a dense network of proteins. Because these microtubules are still trying to push along the microtubules, right? So they generate stress in the system. So if you think about so then there's a instability occurs in this case where you generate large scale flow. So that's a sort of hydrodynamic instability in this case. So this second example here is a dense system. Yeah.
Hydrodynamics is also.
That's right, exactly. So in this case, because these motor protein systems are not polar and not self-propelled, so you're just generating stress. So in this case, the hydrodynamic interactions is the key to observe large-scale phenomena or flow phenomena. In this case, what we're seeing here, the large-scale flow looks like turbulence. People call them active turbulence. Of course, that's not real turbulence, but the point is, again, is because of the nonlinearity in the dense interaction between the constituents.
Is in big flow.
Yeah, it's in the whole.
It's the microtubules are.
Yeah, they're in a fluid, and then they will generate flow, and then there's large scale flow where the microtubules are also flowing with the fluid in the environment. Yeah.
Also, you said the fluid activity is there. Activities. So introduce into that fluid flow field of the fluid.
So in this case, just ATP, which are consumed by the proteins, right? In a chemically active system, you would have the chemicals in the water. If that's fully consumed, then you wouldn't have any. Yeah.
Interactions also contribute to that.
Yeah. So the hydrodynamic interactions is sort of what leads to these large scale behaviors. They don't contribute to activity. Yeah. Any other questions? Yeah.
We said hydrodynamic interaction was there. Right? So hydrodynamic interaction is responsible for the activity. You say.
No. Sorry, could you say that again?
If the hydrodynamic interaction is also responsible for the activity.
No, sorry, that's not. I think that's not what I said. What I said is, so the hydrodynamic interaction are always there if you're in a wet system, right? If you have multiple particles in the fluid, that's always there. However, the large scale behavior of this kind of systems is different from a system. You have water and you have just passive colloidal particles in water, and there's also hydrodynamic interaction. But in this case, because you have motor proteins that are actively trying to slide along the microtubules, you're generating active stress in the system.
No system.
Yeah.
The flow field, then we can say that hydrodynamic interactions can cause the self-propulsion of the polymers. Active.
If you just have regular polymers in water, no, that's not active, right?
That's basically introduce activity.
Sure. If you have an active polymer, for example, the polymer has a head. That's a swimmer. That's a particle. Then yes. Yeah. If it's a regular polymer, then no. Yes. Yeah. So there's sort of two things. One is how do you get your activity or self-propulsion. The other point of this is when you have activity, and then when you couple that to physical interactions, that leads to new collective phenomena which you don't observe in a passive system. So these interactions, for example, the exclusive volume interactions and attractive interactions. We have that in passive system as well, right? But the difference is now you don't have activity. So the key thing is when you couple that to activity, you get this new phenomenon that we see, whether in biology or in these experimental systems. Other questions, comments? Okay, great. So let's look at this video here. It's not, okay. Okay, so what you see here, so this is a experimental video, and you see this defect that's forming in a dense active sort of microtubule solution. So that these defects look very much like what you see in classical liquid crystal systems. And the only difference again here is because now you have additional active stress in the system, and later on we will actually make use of classical liquid crystal theory to understand how this kind of system behaves. And the only new thing really in this context is to add the active stress that these systems are generating. Yeah, question?
What is time scale when the pattern is changing?
This
scale.
I think it's in minutes, if I remember it correctly. Yeah, it's within about 10 seconds or so. Yeah. So it's quite, in this case it's quite dense, so the activity is quite high, so things change around quite a lot, and the defects actually they're evolving and interacting in a quite dynamic way.
Motion is non-linear.
Yeah, this is very nonlinear. So if you do a linear stability analysis, you can't get it. This is a very nonlinear behavior in this case. So you can do other things with these systems. So I've said that if you look at these two microtubules connected with the motor proteins, this aggregate is not self-propelling, right? They can only generate active stress. But what you can do is you can encapsulate these microtubule networks into a droplet. So on the left-hand side, okay, I have droplets, and within each of these droplets, there are these microtubule solutions which will cross-link and form these networks. On the right, there's no activity. So what you see is on the right, the bubbles are just stationary. But on the left, actually, if you think about these bubbles as an active particle, they're actually self-propelling. And in this case, that's because of the stress generated in the system. And if you do a proper fluid mechanical treatment, you're going to discover that these active particles, so this composite system actually is polar now, even though the individual microtubule networks within these droplets are not. So that's also a very interesting example of microtubules. Any questions about this system? Okay, good. So let's think about a second system, which is perhaps the most common that you see in the literature. These are swimming microorganisms. Okay, so these microorganisms comes in various shapes and forms. So here's a few examples here on the left. So you have paramecium, and you have sperm cells, E. coli, and all other kind of cells. So here in the middle, this is a video of the swimming motion of, these are human sperm cells. Okay, so these are active particles in a fluid. And on the right-hand side here, this is a video of a paramecium. What you see here is you have a cell body, okay? And you have these a lot of tiny little hair-like structures on the cell surface. So those are called cilia. So these cilia are actively beating, which will generate a local fluid flow around the surface. So by a mechanical balance, this particle has to self-propel. So essentially, the propulsive force generated by this flagellum is balanced by the drag force that you get as the cell body moves forward. So later on, again, we'll talk about the details of their self-propulsion. But this is, again, a second common system that we use in active matter studies of living systems, which is swimming microorganisms. So the third system that's sort of most convenient if you're an experimentalist is chemically active systems. So these are, for example, Janus particles. So what you have here in this example is you have a plastic bead, which is the simplest way that you can make these guys. And you coat only 1/2 of the sphere with the platinum. So the platinum is a catalyst for the decomposition of hydrogen peroxide. So you then put these Janus particles in a hydrogen peroxide solution, and that's it. That's all you have to do. And what happens here is, because the chemical reaction is only promoted on one side of the sphere, and there's a chemical concentration asymmetry, and that imbalance causes self-propulsion. So on the right here is an experimental video, and what you're going to see is so at the top, in the top row, this is a control experiment, okay, and that means it's not Janus particles. And in the bottom row, these are both of them are Janus particles. And if you look at this vertically, in the first column, I'm putting these particles. One is just regular plastic spheres. One is Janus particles in water. In the second column, at the top, that would be, regular spheres, spherical particles in hydrogen peroxide solution. At the bottom right corner, that's Janus particle in hydrogen peroxide solution. So that's the one we want to look at. So these are just a single particle. And what you see is all of them are moving around. And the reason that these guys are moving around is just due to Brownian motion. And you can clearly see that. So these experiments are synced in time. So what you see is this guy has moved to a much further distance. So yeah.
I can sound simple things which I work at.
Yeah.
Chemical reaction is taking.
Yeah.
So the chemical energy some form of energy is being created by this thing. Energy, not what energy.
So in this case, the simplest way to think about it is essentially there's an osmotic pressure imbalance on the sides. Because the density of, for example, hydrogen peroxide is different. Yeah.
Interacting with the metal.
Yeah. So yeah.
It is the heat energy which is generated by the chemical.
In this case, it's not the heat. No.
What form of it?
It's just you can if you look at the microscopic level, you can sort of
Not the force which is pushing. It's some form of energy.
It is a energy interaction, but so if you look at the energy around the sphere, you will be
What form of energy it is?
Sorry.
What form of energy it is? In chemical reaction.
Yeah.
If you compound this form and some energies either released or absorbed.
Yeah.
It's so thermic in the term.
Yeah.
So I just want to know, I have heard this.
Yeah.
So what kind of energy is created when hydrogen peroxide is interactive with the metal?
Yeah, that's an interesting question. I'm not sure.
How it is converting, keeping the motion.
Yeah.
It is jet motion, something is going.
Yeah.
Pushing back.
I'm actually not sure. So, yeah.
How it is pushing straight way, pushing tilted way.
Right. Yeah.
So this is simple things.
I'm actually not sure. The best way to describe that, to be honest. Yeah. But later on, we'll talk about sort of the
Discuss.
Yeah, exactly. That's a great question. Any other questions about this?
But if there is a chemical reaction which is taking place, we should have a reaction equation. Do we have any equation corresponding to this?
For which one?
The H2 hydrogen peroxide thing.
Equation for
Equation chemical.
So I guess for this system, it doesn't have to have a chemical reaction, right? Yeah.
Means that there's no chemical things.
There is reaction, but the point is you can make a different system where
which compound is reacting with which, and what is the product?
So it's just the hydrogen peroxide is decomposed on the side into water and oxygen. That's it. Yeah, that's it. So
this is happening because of the this platinum coating of the.
Yeah. So it's asymmetric. So it's a diffuse euphoretic motion. Yeah.
Platinum
Yeah.
Hydrogen peroxide is decomposed.
That's right.
That is the chemical reaction which is coming.
Yeah.
Is actually creating some sort of density gradient.
Yeah. Because on this side you don't have that reaction happening. Yeah.
Less dense system.
Yeah.
On this side there's a more dense.
Yeah.
It's a gradient which is
That's right. Yeah.
Which is driving the big.
Driving the particle forward. Yeah. So you don't have to have a reaction, right? The point is, so long as you have a density asymmetry in a diffuse euphoretic system, you would also have self-propulsion. You don't have to have a reaction. So that's the point of these systems. Yeah.
Recent gradient is on extra mains temperature gradient is created in codes, so that
that's right. Yeah.
Or some chemical energy is created at that chemical energy.
So that's driving sort of the from the mechanical. Yeah.
So I don't see how pressure gradient is created because each S2O2 particle is being converted into two S2O plus oxygen particles.
Yeah.
So because of this on this side.
Yeah.
So number of individual particle density of the particle is more.
Yeah.
This side we have a higher side, but it is not yet.
Yeah.
So more particle on this side.
That's right. That's right.
There's a pressure gap.
Yeah.
This side is it?
Yeah. Yeah. Mechanically, that's what's happening, right?
Yeah. Either oxygen is coming out.
Yeah. Yeah. Well, oxygen is coming out. That's right. Yeah.
Okay.
So it's essentially just the density or the concentration of hydrogen peroxide that's different here. You don't have to do this reaction. You can make other kind of diffusive phoretic motion so long as there's a concentration difference, because the interaction energy between the particles, it's the same particles, but the density on the right hand side is higher or lower, so that creates an asymmetry in the system.
It's really density.
That's exactly. That's a diffusive phoretic motion. It's driven by a gradient. No. Yeah, we don't need it. You don't even have to have a reaction for this. Yeah. Other questions? Okay, great. So this is an example of a chemically active system. So you can make other kind of active systems. In this case, these are little robots. So they're about centimeters in length. And so in this case, they're putting into a disk chamber. And what you see is, so in this left system, the number of robots are few. So you see they're more or less in a kind of gas-like phase. Whereas on the right-hand side, you increase the number of these robots. What you see is these guys are sort of accumulating on the surface. There's a surface aggregation of these little robots. So that's an interesting phenomenon, because if you think about a system in thermal equilibrium, the density will be uniform. In this case, there's boundary accumulation, which is actually a key signature of activity, which we'll talk about in the next lecture in developing a model to describe that. Okay, so we've looked at a few examples of systems that one can use in experiments to understand these systems. Here's an example application of what we can do with these active particles. So that's in the context of micro or nano motors. So here's an example of an endogenous enzyme-powered Janus platelet micromotor. So we talked about the Janus particle before, where you have a reaction that creates a density gradient. So in this case, of course, you cannot put hydrogen peroxide solution into the human body to make a micromotor, right? So what people have thought about is, can we make use of natural enzymatic reactions that are happening in the biological system to make a self-propelled particle? So that's what they did here. So this sort of particle here is a platelet cell, and they've coated this with an enzyme. So that's these bluish things here. Again, this enzyme are asymmetrically coated on the particle. And then this enzyme causes a reaction from urea, the decomposition of urea, and again creates a density gradient around the particle, which will allow these guys to self-propel. And what you can do with this kind of systems, you can also make an aggregate of a DOX and this micromotor. So, and then you can sort of guide these guys to a cancer cell. So this DOX will sort of attack the cancer cell. So that's a image from the experiment. This is an in vitro experiment. So that's an example of using micromotors to do delivery tasks in the context of biomedicine. And on the right, there's another interesting experiment. So we've seen the motion of motile bacteria, right? So there's a type of bacteria that's particularly interesting, which is magnetotactic bacteria. So these bacteria contain biologically synthesized magnetic allocrystals within them. And that's very interesting, because then these bacteria actually respond to magnetic field, so which is very useful for medicine, because we have a lot of magnetic devices in the hospital, so we can directly use those systems to control these bacteria. So this is an experiment where people have made sort of 5 microfluidic chambers, okay? And then here what you have, the red is the magnetotactic bacteria, and the blue You guys can't see this. So the blue is sort of the cancer spheroid cells that are done in an in vitro experiment. So what happens here, if you look at the graph at the bottom, so because you can guide these swimmers using magnetic field, so here's on the left, there are 3 different types of field that you've applied to the system. And the last one is the control experiment. And let's look at the control experiment first. So there's 2 columns. One is what's called the on target. So the target is the center cell. And what we want here in this experiment is to guide these bacteria to the center cell. All these 5 cells are connected in a system, but the multi-bacteria is in the system. They can move everywhere, but the point is, can we guide them to the target? So if you do a control experiment, what you see, of course, is the number of bacteria in the on target in the middle versus other chambers are roughly the same because there's no bias in these chambers. Whereas in the left, we've applied a rotational magnetic field, which is the magnetic field profile is designed such that the particles are sort of being directed to the center. And you see that in all 3 cases, the number of bacteria is higher. And what's more important is you can also add what's a selection field, which is essentially a magnetic spatial gradient. And that will allow you to further promote the accumulation of bacteria in the on-target cell compared to other cells by designing the spatial variation of the magnetic field. So that's an interesting application. Okay, so another interesting example is in doping classical materials with active particles. So here we're looking at a gel system. If you think about a gel system, such as a colloidal gel, you have 2 particles that has short-range attractive interaction that's happening. So if you look at these 2 particles which are close together, okay, they're tied together by this attractive interaction. If you put an active particle there, so the active particle is self-propelling, right? So if you think about their force of self-propulsion, so if that force is higher than the force due to this potential, you're going to break apart these 2 particles. If your force is high, you break them apart. If the force is small, of course, you can't break them apart. So there's a critical force where you can compare the self-propelled force with the force due to the energy. If that's comparable, that's a critical value. So if you do a simulation here, there's 2 parameters here. This Peckley S, that's essentially the swim speed, or how active are these guys. And this Peckley R here, that's a rotational Brownian motion, which we'll talk about later on. But what you see here, essentially the point is, by only adding a small fraction of these active particles, you can dramatically change the behavior of a gel system. So what you see here on the right is a phase map here. You have, so when the activity is very low, you have a thermal coarsening behavior. And what you can do actually at high activity is you can induce additional attractive interactions among these passive particles due to the presence of other active particles. Or you can also induce repulsive interaction among these passive particles depending on the Peckley R. So that's a very interesting phenomenon. And here's a second example in the same context. You put a small number of active particles in a gel kind of system. And in this case, if you can do a process where initially you turn on activity, okay, of these active particles, and it's gonna push around these passive particles and try to reorganize them. And after some time, you can turn off activity, and then you can sort of push this gel into an interesting new state. And what happens is if you measure the stress as a function, stress versus strain in this system, what you see is as you increase the self-propelling force or increase the activity of these particles, you can induce a brittle to ductile transition in these materials. So that's an interesting example in a materials science kind of application. So we've seen quite a number of examples today, and now let's take a few minutes and then try to start build some models for it. And we'll start from sort of equilibrium ideas by first talking about Brownian motion. So I'm going to switch to my iPad for a sec, and then we're gonna start writing down some notes for actual theoretical models, all right?
Slow the down counts.
When should I stop the lecture? Is it 1:10 or?
Yeah, 1:15.
1:15, okay, so we still have some time. All right. Okay, so let's first talk about Brownian motion. Okay, so let's think about a classical problem, which is passive Brownian particles settling under gravity, right? We have particles in a dilute regime that is settling due to gravity. So there's a gravitational field. Due to gravity. And so if you think about a particle, right, under the influence of gravity, the velocity due to gravity can be written in the following way. So the velocity would be the force, the gravitational force, divided by a friction coefficient, okay? Let's call that psi. So this is a simple relation. So this is the gravitational force. And psi is the friction coefficient. Okay, so sometimes we also call that a resistance. So in the context of colloidal particles, we'll talk about how do we get this friction coefficient when we talk about hydrodynamics. For now, let's just take this as a sort of phenomenological model. So we can calculate the
particle which is suspended in some sort
of water. Yeah.
So there's a friction. When you just settle.
That's it. So particles settling in water and then under the influence of gravity. So we have a friction coefficient, okay? And sometimes we also call this 1 over psi as a mobility. That essentially tells you if you apply a force, in this case a gravitational force, how much velocity you get. You multiply the force by the mobility, okay, to get the velocity. Okay, great. So let's define a quantity. So we can characterize the system by looking at the number density of particles in space as a function of space, x, potentially time as well. So let's call that the number density of particles. In the system, okay, because we have particles settling due to gravity, there's a flux associated with their velocity. So we can write that down. So the flux due to gravity would be, let's use symbol J for flux. And I'm using 1 underscore for a vector, just for the notation. So the flux then would just be the velocity due to gravity multiplied by the number density, right? So that's the velocity times the number density, okay? Great. But we also have Brownian motion, which for now we're going to model that as a diffusion. So diffusion, which is in this case governed by Fick's law. So that's a constitutive relation between the flux due to Brownian motion and the particle density gradient.
That's completely cool.
So we can write that as a diffusive flux.
I think this Jg, that is the that's current.
Yeah, that's the current or the flux. Yeah.
The velocity times
the density of the particle. Yeah, that's right. So we also have a diffusive flux, which from Fick's law, we write that as a, minus the diffusivity times the density gradient, okay? And here, that's a diffusion coefficient. So to be more precise, as a translational diffusion coefficient. Translational diffusivity. Okay, so that's great. But we also have, when we think about these systems, we can also write down a conservation law because the number of particles is conserved in the system. So we can write down a conservation law for the density, right? So in general, the conservation law is written in the following way, right? The time derivative of the conserved quantity plus the divergence of the flux has to be equal to 0. So that's the net flux or the total flux is equal to 0. So in this case, we have two fluxes. One is the fact that there's a gravitational force that induces a flux due to the gravity, and the second force is due to the diffusion. So if we add them up, we would have the total flux. One is due to the gravity. The other one is the diffusive flux. Okay? Any questions about this? Okay, great. So let's think about steady state, where we just get rid of the time derivative, and then at steady state,
Yes, sir.
we have the divergence of the flux vanishes. Okay? So that's one thing we can think about. Okay, in Professor Mishra's lecture, we'll talk about thermodynamic equilibrium. So that's a much stronger condition than steady state. So if we think about thermodynamic equilibrium, what we have is we can have a net current or we can have a net flux. So which means that the total flux is 0, right? Okay, that's great. And we're thinking about gravity. We can also write the gravitational force as a gradient of the gravitational potential. Okay? And we know also from equilibrium that the density distribution in the presence of a field like this has to be Boltzmann, which means that the number density goes like a constant times e to the minus the potential divided by kBt, right? Okay, that's from thermodynamics. So let's think about this. At equilibrium, we have our total flux, which is one is due to the gravitational force, the other is due to Brownian motion, that's equal to 0. But what we can do is, let's make use of the equilibrium distribution and to figure out a relationship between the diffusivity and the thermal energy, kBt. So let's do that. So the first part, that's the velocity times the density, which becomes minus 1 over zeta. The force, okay? The force is minus of the gradient times n. So that's the first part of the equation. The second part is jd, which we have from before, minus dt grad n. Okay, so these add up to 0. So let's carry out the calculation by taking the equilibrium distribution. So let's take the gradient. Okay, so that's just, if I take this equation here, take the gradient, I have c minus grad vg over kBt times e to the minus vg over kBt, right? So that's just equal to minus grad vg over kBt. So notice that because I'm taking the gradient of an exponential function, you get the exponential out. So that's just n itself. Okay, so we can write it in this way. Okay, any questions about this? Just doing some manipulations. Yeah.
So it's like that also actually have not common physics.
Yeah.
Well, this one thing is very in upper density. So that we equilibrium probability.
It is it's essentially a number per volume. Yeah, the way to think about it is it is a probability distribution of where particles are in space. Yeah. Which we'll talk about later on in more detail, how to construct that kind of a probability for active particles. But yeah, that's correct.
Yeah. Realizing that this is a flux like this probability takes a beam to affect velocity erosion.
Which one? The first one here?
This is the number is right. And we're multiplying.
Yeah. So that's the flux due to a systematic velocity in the system. That's a constitutive relation. Yeah.
Is it 6?
Fixed law is for the diffusion. That's the second part. The first part is just due to the fact that there is systematic motion in the system due to external force. So the flux would just be the velocity times the quantity that's conserved. That's the definition of a flux due to external field. Any other questions?
That's good.
Okay, great. So let's carry out this calculation. And so what we have discovered is 1 over psi gradient of the potential times n, right? That's the first part of the flux. plus the second part, which is minus dtn, and then that becomes plus dt grad vg over kbt n. That's equal to 0. Yeah, there's a sign error here. So there's a minus sign because the force is the minus of the gradient of the potential. So that's equal to 0. And then what we have here is the following: 1 over psi times the gradient times the density is equal to dt over kbt gradient vg times n. So notice the common factors here, right? So what we have is, we have a relation that says that the diffusivity is kbt divided by psi. So that's called a Stokes-Einstein relation. So a few comments about this. So more generally, the friction that we talk about, the friction psi doesn't have to be an isotropic quantity, can be in general a tensorial quantity. We call that a resistance tensor or a friction tensor, if you will. So we use R, for example. And so in this case, we're looking at, so sometimes we give it a subscript that tells us this friction relates the velocity to the force, because you also have rotational friction that relates the torque to the angular velocity, which we'll talk about in just a moment. So the frictional tensor here is in position space, so which essentially couples the velocity of the particle to the force, to the external force. So what we can say is, then the inverse of this tensor is the mobility, right? So that's RFU to the inverse. So this subscript is interesting. The way I'm writing it this way is because if you can think about the external force, or the other way, if you think about the velocity that you get from the external force, then you multiply the force by the mobility, right? So you multiply the mobility tensor. So I'm using 2 underscores now to denote a second rank 2 tensor. So that's MUF times the external force. So that's a typical relation. The reason that we have this kind of linear relationships, again, we'll talk about later on when we talk about hydrodynamics. So the way I'm writing this is the following. So you can view this subscript as a form of sort of tensorial contraction, right? So if you have MUF times F, so these are contracted, then you get a velocity out of it. In the inverse relation would be the force is the resistance times the velocity. So then this U sort of contracts with the U on the right, and then you get a force out of that. So that's just a conventional notational convenience in terms of resistance and mobility tensors. Any questions about this?
Okay.
So for a sphere, U is the velocity that you get if you apply a force on a particle. What is the velocity that the particle get due to the force? So that's U, and for F is the external force that you've applied. So if you think about a spherical particle, we would have the resistance tensor to be isotropic because there's no directional bias in a sphere. So that means we can write that as psi times the identity tensor. So that's identity. Okay, great. So a few remarks about the relationship, and then we'll stop our lecture today. So we talk about the first relation, which is, we have a gravitational force, and we apply the force, and the response of the particle is it gets a velocity. So then this psi characterizes the response of the particle to an external force or an external field. So in this case, more specifically, that's due to friction. And the other thing is we talk about diffusion. So dt characterizes thermal motion of the solvent. In this case, it's just water molecules. Notice one thing, that the friction is also coming from the solvent. So is the diffusivity. So they're of the same origin. And what this means is the Stokes-Einstein relation which connects these two quantities, right? That is a special case of what's called a fluctuation dissipation theorem. So the fluctuation is the thermal motion of the solvent, and the dissipation is the friction, the psi, the friction coefficient. So I guess we'll stop here for this lecture, and then we'll come back in the afternoon to talk about building up this model for active particles. Any questions before we stop? Yeah.
So suppose we imagine a system where there is no velocity, so there is an external force, which, for example, a liquid crystalline system might be, which is just which are moving the particles anyway, but they're just orienting the particles.
So you want to have an orienting external fields. Yeah. And what's the question?
Consider that as an active system.
How do you orient the particles, for example?
Which is the
so you just have a torque, for example, that's spinning these particles.
Torque.
Due to the fluid or some external field?
No, some external.
You could consider that as an active system. It depends on the context. So you do have these sort of roller systems where they don't self-propel in physical space, but they're spinning. But because if you're in a wet system, if you're in a fluid, the fluid at the particle surface often has to satisfy a no-slip condition, right? They have to follow the motion of the particle. Then you generate flow in the fluid. So that's also one example of an active system. But then it's driven. So I guess the point is, in this system, sometimes there's a sort of a continuous spectrum of what is driven and what is a active system. It's not as clear cut as one would have hoped. But the point is, even for the system you proposed, you can use the tools that we are developing to model them. So from a sort of utility point of view, then it's useful to treat them that way. And people have done those kind of experiments as well with these rollers. Yeah. Any other questions?
So
okay, we'll come back 3. Okay, let's check.